I am Judith Mano Gracelyn from Physical Science Department. Today I am going to present a topic about chemical bonding and biocoordination compounds. Let's see about chemical bonding. Chemical bonding. A chemical bonding is a lasting attraction between atoms or ions that enables the formation of molecules, crystals, and other structures. The bond may result from the electrostatic force between oppositely charged ions as in ionic bonds or through the sharing of electrons as in covalent bonds. The strength of chemical bonds varies considerably. They are strong bonds or primary bonds, such as covalent, ionic, and metallic bonds, and weak bonds or secondary bonds, such as dipole dipole interaction, the London dispersion force, and hydrogen bonding. How does a chemical bond occur? Every atom is composed of a core with positively charged protons and neutral neutrons, and it is surrounded by an outer layer called the electron cloud, which has a negative charge. Opposite charges attract both within the same atom and between atoms. This attraction forms chemical bonds between different elements. Atoms round out their electric charges by electron exchanges. They can give up, accept, or share these particles to achieve stable electronic configuration, meaning a lower energy consumption. Characteristics of chemical bond. They hold atoms together inside the chemical molecules. The strength of a chemical bond is determined by the difference in electronegativity. Generally, the numbers of electrons are even. Covalent bonds or exist as an gas, solid, or liquid. Some covalent bonds are soluble in water, while others are in acid. Covalent bonds conduct electricity in the presence of an aqueous solution. Ionic bonds have high melting and boiling points. Metal bonds are good conductors of heat and electricity. They are typically in a solid state and they are highly malleable. Now let's discuss the types of chemical bonds. Covalent bond, ionic bond, and metallic bonds. Covalent bonds. Covalent bond, it occurs when non-metallic atoms share electrons in this type of bond. Electrons move between atoms producing polar covalent bonds. In ionic bonds, this occurs when metallic and non-metallic atoms bond and an electron charge is given from one to the other. As a result, both negatively charged ions and positive charge. In metallic bonds, these are formed between atoms of metals whose atomic nuclei gather and are surrounded by the electrons like a cloud. This is a strong kind of bond. This is spread out like a network. Lewis theory. American chemical physicist Gilbert Lewis devised the octet rule in 1917. It explains how atoms of different chemical elements combine to form bonds. This theory proposes that the ions of elements on the periodic table fill their lost energy levels with 8 electrons. This way, the molecules achieve stability at the level of their electronic structure. Thus, elements with high electronegative charges gain electrons until reaching the octet, while those with low electronegativity usually lose them to achieve the same goal. Lewis' theory of chemical bonding. An atom can be viewed as a positively charged kernel and the outer shell. The outer shell can accommodate a maximum of 8 electrons only. The 8 electrons present in the outer shell occupy the corners of a cube which surround the kernel. The atoms have an octet configuration. Example, 8 electrons in the outermost shell, thus symbolizing a stable configuration. Cosel's theory of chemical bonding. Noble gases separate the highly electronegative halogens and the highly electropositive alkali metals. Halogens can form negatively charged ions by gaining an electron, whereas alkali metals can form positively charged ions by losing an electron. These negatively charged ions and positively charged ions have a noble gas configuration, that is, 8 electrons in the outermost shell. The general electronic configuration of noble gases is given by Ns2 NP6. Biocoordination compounds. Coordination compounds play an important role in many biological processes in plants and animals. Names and functions. Name: hemoglobin. Its function is to transport and storage of oxygen. Chlorophyll. It functions to photosynthesis. Ferrodoxins, rubridoxins. The functions of ferrodoxins and rubridoxins is electron transfer. Hemoglobin. Hemoglobin in the red blood cell carries oxygen from the lung to the tissues. It delivers the oxygen molecule to myoglobin in the tissues. When the oxygen has been released for cell respiration, hemoglobin loses its bright red color and becomes purple. It then combines with the waste carbon dioxide produced by the cells and deposits in the lungs so that the gas can be exhaled. Chlorophyll. Chlorophyll is a magnesium propane complex. The magnesium is at the center of the modified propane ring septon. The oxidation state of magnesium is +2 Mg2+. The modified porphyrin acts as the ligand. There are several kinds of chlorophyll that vary slightly in their molecule structure. In plants, chlorophyll A is the pigment directly responsible for the transformation of light energy to chemical energy. Hence, in plants, the green pigment chlorophyll helps photosynthesis, the conservation of atmospheric carbon dioxide and atmospheric moisture into carbohydrate and molecular oxygen in the presence of sunlight by the plant is called as photosynthesis. Chlorophyll acts as a light sensor in this important process. Thank you.